like to present today the way of increasing the engraftment capacity and the function of hematopoietic stem cells after transplantation. As we know, stem cells are the only one cells in the organism which can self-renew and in the same time differentiate towards other types of the cells. Hematopoietic stem cells, which residue in the bone marrow, are responsible for the process of hematopoiesis, in which from the hematopoietic stem cells we can obtain the cells of the lymphoid lineage as well as all the cells from the myeloid lineage. So we are talking here about all the granulocytes, monocytes, platelets, and erythrocytes. Hematopoietic stem cell is the cell which is also responsible for the renewing the process of hematopoiesis after all the cells are destroyed during the myeloid or immunoablative procedure, which is the procedure used mostly for preparation patients for the transplantation. Hematopoietic stem cell is differentiated by the number of markers. It's mostly CD34 positive, as well as CXCR4 positive, CD20 positive, CD90 positive, CD17, which is known as a c-KIT. The marker CXCR4, known as well as a CD184, is the transmembrane receptor, which is present not only on hematopoietic stem cells but also on all hematopoietic progenitor cells. It is receptor for SDF1. SDF1 is a stromal derivative factor 1, which is secreted by bone marrow stromal cell, and it's the strongest hemoattractant for hematopoietic stem cells. So, hematopoietic stem cells, which are CXCR4 positive, migrates toward the SDF1 gradient, and it's forming the axis CXCR4-SDF1. This formation is responsible not only for the migration of hematopoietic stem cells, which we observe in the process such as homing, which is a process during which the cells migrate towards bone marrow after transplantation, but unfortunately also SDF1 is secreted by other organs, and here we are talking about the metastasis process. This axis is also responsible for the stem cell survival, their proliferation, and also for the normal hematopoiesis. Talking about hematopoietic potential of the cells, we are thinking first of all about their potential to migrate towards the SDF1 gradient, but also their potential towards proliferating and renewing the hematopoiesis after the transplantation. This process we can check during the assay of hematopoietic colonies, and in my experiments I was interested in growing colonies from one of the myeloid compartment. I was concentrating on the progenitors of the white cells, which differentiate towards granulocytes and monocytes. This is the example how the colony looks like. So hematopoietic stem cells and progenitor cells, which is the material used for transplantations, can be obtained from bone marrow, nowadays more often from mobilized peripheral blood, but also from cord blood. And a lot of factors such as anticoagulants, temperature, storage time, but also the knowledge of the personnel who is preparing the cells for the transplantation are responsible for cell survival, but also for the ability to proliferate and reconstruct hematopoiesis after transplantation. Although there is no one standardized procedure of storing and preparing these cells among the transplantation centers in the world. So the aim of my study was to evaluate the influence of the most common used anticoagulants, such as EDTA, CPDA1, which is the most used in England, and the heparin, very common used anticoagulant from 1950s. The temperature of 4 degrees Celsius and 37, and different storage times. The cells which were not stored at all, they were stored for 1, 6, 12, and 24 hours. I was checking how these factors will have influence on the migration of bone marrow stem cells towards the gradient of SDF1 and the ability to create hematopoietic colonies. As a material, I used the mice bone marrow and the methods which I was using. So I collected the cells from femur bone from mice, and then I divided them for different groups. Each groups were stored with different anticoagulants, EDTA, CPDA1, heparin, and the control group, no anticoagulants at all. And they were stored during 1, 6, 12, and 24 hours in temperature of 37 degrees Celsius or 4 degrees. I also had the group of fresh collected cells which weren't stored at all. All these groups were moved towards the hematoxis assay, which is the assay which show us how many cells are able to migrate towards the gradient of hemoattractant, in my case SDF1. The cells which we graded, I divided and I made a group of the cells which I was able to check how many cells migrated. I done that by using the flow cytometer. And the second group was used to start the clonogenetic assay. So the first method, hemotaxis. For this method, I've been using the plates, 24 wells, transwell plates. Each well in this plate includes one insert. Insert divides the well in the two compartments, upper and lower. Each insert has the porous membrane which allow the cells to migrate from upper compartment when we put the cells in medium towards the lower where we place the medium with chemoattractant, in my case SDF1. And then we can check how many cells migrated. So all the groups were put into this experiment. This is the movie. In which you can see how the cells are migrating during the hemotaxis assay. These are all the cells put on the upper compartment on the well. And one more, which is showing the single cells now, which is migrating towards the gradient of chemoattractant. So with the pipette, we are placing the chemoattractant, and we can see how the cells is migrating towards the gradient of this substance. So this graph is presenting the results of the chemotaxis assay, which was done on the cells which were stored in the 4 degrees Celsius. So here we have the percent of the cells which migrated towards SDF1, and this is the storage time for how much they were stored, all the groups. The single lines represent all the groups which were incubated with anticoagulants, EDTA, CPDA, and heparin, and the control groups, no anticoagulants. We can see that all the factors like storage time and also temperature and types of anticoagulants have influence on the number of cells which were migrating towards SDF1. We see the trend of decreasing and increasing the number of cells which were migrating, and the 6 hours seems to be a peak where the almost 30% of the cells migrated. The more interesting seems to be the 37 degrees Celsius, which we have similar graph, percent of migrating cells, the storage time, and different groups of anticoagulants. Also, the cells are decreasing and increasing towards the time, but also 6 hours seems to be very interesting, where the anticoagulants such as heparin and CPDA seems to be the best. When we compare with 4 degrees Celsius, we can observe that almost 60% of cells migrated comparing to 30% in 4 degrees Celsius. The second test which I performed was a clonogenicity test, where I grew the colonies towards the monocytes and granulocytes. I used the hematopoietic progenitor cells, which I culture in vitro, and I push it to differentiate towards granulocytes and monocytes using the GM-CS factor and interleukin-3. As a colony, I set up the number of cells 50, the group of 50 of cells. Let's look at the 4 degrees Celsius number of CFU-GM colonies versus storage time. So, similar pattern, similar 6 hours storage time, but the number of colonies is not that great. So here we have 37 degrees Celsius. We have 6 hours of storage of the group of cells with heparin seems to be the best condition for storing the cells which are getting ready to be transplanted. It's not only the number of the colonies, it also matters how the colonies looks like. So here we can see on the pictures that cells stored with the heparin during 24 hours, 12 hours, 6 hours, and 1 hour in the condition of 4 degrees Celsius or 47. We can say that 37, the colonies looks much bigger and better, but the best, again, 6 hours, the colonies have the biggest size of all. So after knowing that the heparin, it will be the best anticoagulant, 6 hours of storing the cells, and also 37 degrees Celsius, I was interested to see if the concentration of heparin can matter versus the number of the growing colonies. So I tested 10, 20, 50, 100, and 2000 units per milliliter, and I look how many colonies I can grow. The best, it seems to be the 10 unit per ml, and the same 37 degrees Celsius. I move forward and I ask myself question, where the purest colonies, the pure monocytes and pure granulocytes can grow the best, in which condition? Of course, using now heparin and 6 hours of storage time, I compare 4 degrees Celsius and 37. It looks like the pure monocytes and pure granulocytes colonies grows better in warmer condition. My conclusions. Ability of hematopoietic stem cells migration and the creation of the granulocyte and monocytes colonies in vitro depends on type of used anticoagulant, temperature, and the storage time. Heparin, it seems to be suggested anticoagulant for bone marrow stem cell preservation. Bone marrow cells, which are preserved in 37 degrees Celsius, demonstrated better migration response and proliferative potential towards granulocytes monocytes colonies. Higher migration and also number of colonies I noticed in 6 hours of storage. Number of migrated cells and colonies was changing periodically during 24 hours bone marrow storage. And I think that the stem cell migration capacity towards SDF1 should be the standard test in transplantation material quality evaluation. Thank you for your attention.